Hello everyone, welcome back to JDC Classes English. So dear students, as you all know, MRB Pharmacist vacancy for Tamilnadu state is there, and for this preparation of this post, Master Stroke series, one of the series that has been started by JDC Classes English, where you are going to have 30 question videos with a particular topic and its detailed explanation from expert faculties. So today we are going to have biochemistry and clinical pathology, and the topic of today's subject will be introduction to biochemistry. So from this topic we are going to have some of the important questions we will be discussing in today's video. So as we all know that today's topic is introduction to biochemistry. Let's get started with our first question. So the first question here is the term biochemistry first coined by. The term biochemistry first coined by. Biochemistry word was the first coined by. It was Carl Newberg in 1903, Horace Wells in 1903, Gardner Quincy Colton in 1903, or was it William T.G. Morton in 1903? So, if we talk about biochemistry, Carl Newberg, he is also known as the father of biochemistry, father of modern biochemistry or biochemistry. So, it was the first person, Carl Newberg was the first person who coined this word biochemistry. Okay, next Horace Wells. He was the person who first used nitrous oxide in dentistry. Gardner Quincy Colton was the person who discovered, who first talked about that N2O, that is nitrous oxide, can be used in the as an anesthetic agent. William T.G. Morton, he was the person who used ethers as an anesthetic agent. So Carl Newberg will be the right answer. That is A will be your correct answer from this question. So the term biochemistry was coined in 1903 by a German chemist named Carl Newberg. Okay, let's get with our second question, that is cell term was given by. The cell term was given by, was it Robert Hooke, Topham, Swan, or Dewberry? So as we know that cell term was first coined by Robert Hooke. It was Robert Hooke who studied the cell in dead cells, dead cork cells. So Robert Hooke will be the right answer. Robert Hooke will be the right answer. Cells are the structural and functional unit of a living microorganism or living organism, and Robert Hooke was the person who discovered this cell in 1665 via the self-prepared microscope. He was the person who first prepared the microscope. Okay, so for this MRB pharmacist preparation, Tamil Nadu, we are conducting test every day at 6 p.m. and it's totally free of cost. Nothing charge will be there. How to join this test? You simply have to visit Google Play Store, where you download the JDC Classes app and you just simply visit the batches section and click the code MRB25 and you will be joined in this batch. So in this slide you can see that JDC Classes you can visit on the JDC Google Play Store. So you simply install this application, then you submit your detailed informations, and then you will be shifted to some of this interface where you simply click on the batches and click on this plus icon, where you will be asked a batch code and you have to write it down MRB in small case 25 without any space and simply press OK. Within 2 to 3 hours you will be joined in a batch, in a new course batch, where you can have this free of cost test series. Okay, so let's have our next question. What is biochemistry? What do you mean by biochemistry? Study of chemistry of human body, study of chemistry of carbon compounds, study of chemistry of living things, study of all matter. So if we talk about study of chemistry of carbon compounds, it will be organic chemistry. Study of chemistry of human body. So study of human body can be also termed as biochemistry, but the exact meaning of biochemistry is bio, that means life, means something that is living. So study of chemistry of living things, this is our most appropriate answers. But if this option is not present in your options, then you can have this option as the correct one. But here we will be writing it, study of chemistry of living things will be biochemistry, will be termed as biochemistry. Okay, so biochemistry can be defined as chemistry of living cells. The chemistry of living cells, which is the structural and functional unit of the living body. What is the structural and functional unit of living body? So living body's structural and functional unit will be tissue, cells, organ systems or organs. So a cell combines to form tissue, tissue combines to form organs, and organ combines to form organ systems. So cells are the basic structural and functional unit of any living body. So right answer will be cells. B will be the right answer. B will be your right answer. The cell is the structural and functional unit of a living body. Many cells group together to form tissue, many tissue group together to form organs, many organ group together to form organ system, and many organ system group together to form coordinate to form a living body. Okay. Look at the next question, which is called as a powerhouse of cell. What do you mean by powerhouse of a cell? When we talk about powerhouse of a cell, so there are many cell organelles. Inside a cell, there are many cell organelles like nucleus, mitochondria, endoplasmic reticulum, and lysosomes. So lysosomes contains hydrolytic enzymes that are also known as, lysosomes are also known as suicidal bags. Endoplasmic reticulum, they are mainly of two types. It will be RER, that is rough endoplasmic reticulum, and soft endoplasmic reticulum. RER contains ribosomes. They contain ribosomes, and they are basically used in protein and lipid synthesis. Mitochondria is related to energy formation. Krebs cycle basically occurs here. That is related to ATP formation. That will be also known as power generation, and hence mitochondria will be known as powerhouse of a cell. Nucleus, what do you mean by nucleus? So nucleus are the substances, nuclear are that cell organelle that contains hereditary, or you can say genetic material. They contains genetic material. So right answer will be mitochondria. That is B. This mitochondria was first discovered by a physiologist known as Albert von Kolliker. Mitochondria is double membrane system that is having outer mitochondrial membrane and inner mitochondrial membrane. Inner membrane contains a series of folds known as mitochondrial cristae, and internal chamber of mitochondria is referred to as matrix or mitosol. The matrix contains several enzymes concerned with the energy metabolism of carbohydrates, lipids, amino acids, e.g. citric acid cycle, that is also known as Krebs cycle. This Krebs cycle occurs at the matrix. This matrix basically contains various enzymes that are required for the Krebs cycle, or you can say citric acid cycle, and beta oxidations. So this is all about our mitochondria. Next question, the plasma membrane is made up of. Plasma membrane is made up of only protein and carbohydrates, only protein and lipids, protein, lipids and carbohydrates, or protein, some nucleic acids and lipids. So plasma membrane, that is also known as phospholipid. Bilipid membrane with protein, that are also known as integral proteins, and some amount of carbohydrates. So we are having three molecules, biomolecules, that are protein, lipids and carbohydrates. There are two lipid membranes, bilipid system with proteins embedded on those phospholipids, that are lipids, and some amount of carbohydrates for the energy. So your right answer will be protein will be having 55% of content of your plasma membrane, then 40% of the there will be lipid, and 5% of the carbohydrate. So this is a lipid bilayer, one here and one above. It will be like this is lipid, and there will be proteins that will be available for the exchange of matter. Which region of the endoplasmic reticulum ER is involved in protein synthesis? Which region of the ER is involved in the protein synthesis? When we talk about protein synthesis, so protein synthesis basically occurs in the ribosomes. So rough endoplasmic reticulum RER, smooth endoplasmic reticulum, that is SER, both RER and SER, neither RER or SER. So just cut C and D options. Now I have told already that endoplasmic reticulum is basically having two types. One that will be rough endoplasmic reticulum, and the another one is smooth or soft endoplasmic reticulum. So when we talk about rough endoplasmic reticulum. This rough endoplasmic reticulum is having ribosomes attached at the endoplasmic reticulum, and hence this RER is responsible for the protein synthesis. SER is responsible for the lipid synthesis. So RER will be having protein synthesis mechanisms. A option will be the right answer. A will be the right answer. Which region of the endoplasmic reticulum? Okay, all right, okay. Let's get to the next question. Which organelle functions in the synthesis of proteins? Which organelle functions in the synthesis of proteins? So, what is the cell organelle that is responsible for protein synthesis? Is it mitochondria, ribosome, cytoskeleton, or nucleus? So, nucleus is something that is related to hereditary, or you can say genetic material transfer. Cytoskeleton, is it cytoskeleton that is responsible for the protein synthesis? No, not at all. Ribosomes and mitochondria. Mitochondria is related to protein powerhouse generation, like ATP synthesis. And ribosomes, this ribosome is something that is related to the protein synthesis. There are some enzymes. What kind of RNAs are there? So ribosomes contain rRNA. Ribosomes are having rRNA that are responsible for protein synthesis. There are three kinds of RNA: rRNA, tRNA, and mRNA. So ribosomes is having rRNA that further participate in the protein synthesis. So B option is right answer. Also known as ribosomal RNA. Mitochondria, it is related to production of energy, synthesis of ATP, initiation of apoptosis. Next, ribosomes, synthesis of proteins. Cytoskeleton, determination of shape of the cell, stability of cell shape, and cellular movement. Nucleus controls all activities of the cell, synthesis of RNA, sending genetic instruction to the cytoplasm of protein synthesis, formation of subunits of ribosomes, control of cell division, storage of hereditary information in genes in the form of DNA. Next question, who discovered mitochondria? Who was the person who discovered mitochondria? Robert Hooke, Albert von Kolliker, Antony von Leeuwenhoek, or Rudolf Virchow? So the person who discovered cell theories, Leeuwenhoek, the person who basically first coined or first seen the living cells. Albert von Kolliker was the person who first discovered mitochondria. Robert Hooke first discovered cell in a dead cell, cork cell. So Albert von Kolliker will be the first person who discovered mitochondria. Mitochondria, as we all already have studied, that Albert von Kolliker was the person who discovered them. And next, mitochondria are a double member system, like having inner cristae and matrix, and matrix is containing the enzyme that is responsible for the Krebs cycles and all. Next question, which of the following statement is true about the nucleus? Which of the following statement is true about the nucleus? It is present in prokaryotes. So true, you have been asked the true statements and nucleus. So nucleus, is it the prokaryote that contain nucleus? No, not at all. Prokaryotes are primitive organelles or primitive cells, and these kind of organelles do not contain any kind of DNA, rather they contain circular RNA. It is called the brain of the cell. So this is correct, they don't have any, prokaryotes do not have DNA. It is called brain of the cell. Yes, this is the only organelle that controls all the activities that are actually happening inside the cell. So yes, this is the brain of the cell. It contains DNA and other genetic materials. So yes, they are having hereditary materials, DNA and other genetic materials. So all of the options, all of the above are correct. All options are correct, true. So D option will be right. D will be the right answer. So nucleus is the largest structure present almost at the center of the cell. This is nucleus. See the vastness of this organelle. Nucleus is present in all cells in the body except in the RBCs. One of the most important question, that RBC are the cells that do not have any nucleus. RBC do not have nucleus. The cells with nucleus are called eukaryotes, and those without nucleus are known as prokaryotes. Presence of nucleus is necessary for cell division, and it is also known as brain of the cell. The nucleus contains nucleolus and chromatins. Chromatin are the thread-like materials, and nucleolus rough granular structure of the nucleus. Each nucleus contains one or more nucleoli. The nucleolus contains RNA. This nucleolus is having RNA that is responsible for the protein synthesis. Okay, so for the best preparation of MRB pharmacist, you must have go through the best materials, best contents, and for that JDC is going to help you with the new books especially designed for MRB pharmacist. One of them is MRB Pharmacist Examination Success Kit. Then we will be having MRB Pharmacist Examination Success Kit. This will be theory book and here we will be having objective. So you just make sure that you prepare best content from here and practice the best of the questions with that related topics. Further, you will be also having online test series with 200 premium test and 35 mini test with MRB Pharmacist online test series. And you are also having crash courses for online crash course for Tamilnadu Pharmacist. These all you can have at the JDC Classes app. So you simply go to the Play Store and download the JDC Classes app and see all these courses inside there. Which of the following cell organelle is called as a suicidal bag? Which of the organelle is called as a suicidal bag? So lysosomes, Golgi bodies, cell membrane and mitochondria. Suicidal bag. So I already told you that lysosomes are the person that contains some of the enzymes that are responsible for when the cells get degenerated. So basically lysosomes are known as suicidal bags. They contain hydrolytic enzymes that are responsible for the cells that are degenerated, and Golgi bodies packaging, cell membrane to give shape, and mitochondria to provide energy. So right answer will be A, that is lysosomes are suicidal bags. Lysosomes are spherical vesicles enveloped by a single membrane. They contain 60 kinds of digestive and hydrolytic enzymes. Lysosomes enzymes work best at an acidic pH. Lysosomal interior pH is of about 5. Lysosomes were discovered by Christian de Douai, the first person to discover this lysosomes, and they are known as suicidal bags. Acid phosphatase, one of the most important thing. Acid phosphatase is considered as a marker enzyme for lysosome. What? Acid phosphatase. There are many times this question has been asked in the pharma competitive examination, like what is the marker enzyme present in the lysosomes? What will be it? It will be acid phosphatase. Acid phosphatase. Next question we are having, what is the innermost part of mitochondria called? What do you mean by innermost part of mitochondria? Is it nucleus, cytoplasm, matrix or cristae? So the foldings of the mitochondria, since mitochondria is a two-layered structure, the inner foldings make cristae, nucleus, no, matrix. Matrix is the inside, the innermost part of the mitochondria, and this matrix is responsible for various enzymes that are required during the Krebs cycle, or you can say beta oxidation. So C is the right answer, that mitochondria's innermost structure is known as matrix. So look at the structure of mitochondria. We are having one membrane here, and then we are having this. Look, these membranes, the second layer membranes. So these structures like these are known as cristae. These are known as cristae, and the cristae are having matrix. Which of the following cell organelle is responsible for transporting, modifying and packaging proteins and lipids? Which of the following cell organelles are responsible for transporting, modifying and packaging of proteins and lipids? So is it mitochondria? No, not at all. This is related to the Krebs cycle and ATP production, so powerhouse generation. Endoplasmic reticulum, they are related to production, that is synthesis of this protein and lipids. So no, they are not related to transportation, modifying and packagings. DNA, no, not at all. DNA are related to the hereditary materials. So this is also not correct. But Golgi bodies, Golgi complex. This Golgi complex is basically also known as post office of a cell. They are responsible for the what? You can see transportation, modifying and packaging of the proteins and lipid materials that has been synthesized here. Endoplasmic reticulum, RER and SER. So C option, that is Golgi complex, will be a right answer. So you can see the Golgi bodies here. The Golgi apparatus, also known as the Golgi complex, is a factory where proteins from the ER are further processed and sorted before being transported to the final destinations: secretion, lysosomes, or the plasma membranes. DNA is stored in which of the following cell organelle? DNA is stored in which of the following cell organelle? Is it cell wall, cell membrane, nucleus, or cytoplasm? So it is nucleus where DNA is stored. It is the nucleus where DNA is stored. So C option is right answer. I guess this one is very easy. DNA is the primary stored in the nucleus of eukaryotic cells, and we have talked that RBC are devoid of nucleus. In the animal cell, that is eukaryotic and it doesn't have a nucleus, that is RBC. When which of the following cell nucleus is not present? Which of the following cell nucleus is not present? Eukaryotic cell, prokaryotic, both of the above, none of the above. So prokaryotes do not have nucleus, do not have nucleus, not present. When you see the questions that are having not present, not correct, incorrect, not true, please mark it down the question what actually has been asked. So not present, it will be prokaryotes. Prokaryotes do not have nucleus. So yes, this is our right answer. So B will be the right answer since eukaryotic cells have nucleus. B will be the right. Prokaryotes smaller in size. They are enveloped by a rigid cell wall. They do not have subcellular organelles. Okay, nucleus, they do not have nucleus, while eukaryotes they are more organized cell organelles, eukaryotic cells. RNA is present in which of the following cell organelle? RNA is present at cell wall. No, not at all. Ribosomes, nucleus, and cytoplasm. So ribosomes have rRNA that are also known as ribosomal RNA. So ribosomal RNA are present at ribosome that are responsible for protein synthesis. So yes, B will be the right answer. B will be the correct answer. RNA is present in ribosomes, specifically at ribosomal RNA. Ribosomes are responsible for protein synthesis, and rRNA plays a structural and catalytic role like rRNA, mRNA, tRNA. There are three kinds of RNA. rRNA make up the structural and functional core of ribosomes. mRNA binds to the ribosomes to provide the template for protein synthesis. tRNA, that is transfer RNA, help bring amino acid to the ribosome for protein assembly. So this is all about RNA. Locations of ribosomes containing RNA: cytoplasmic ribosomes. Which part of mitochondria contains enzymes involved in energy metabolism? Which part of mitochondria contains enzyme? Where are the enzymes present inside the mitochondria? Outer membrane, inner membrane, matrix, or cristae? So the matrix are locations where these enzymes that are related to energy production, ATP synthesis, are having enzymes stored. Matrix, it is matrix. So the matrix contains several enzymes concerned with the energy metabolism of carbohydrate, lipid, and amino acid. The mitochondria matrix contains large variety of enzymes, including those that convert pyruvate and fatty acid to acetyl-CoEnzyme. This pyruvate that has been formed during glycolysis cycle, this pyruvate enter inside the Krebs cycle via this acetyl-CoEnzyme A formation. A cell organelle that is present in animal cell but not present in plant cell. So plant cell do not have this kind of organelle, but animal cell have, like cytoplasm, centrosome, mitochondria, or it is cytoplasm. So cytoplasm present on both the cells. Mitochondria is having both the cells. Centrosomes and cytoplasm. Cytoplasm will be having both. Okay, so we are having two cytoplasm and centrosome. This centrosome is present only at animal cells. Centrosomes do not present in the plant cell. Centrosomes. Same like if we talk about the plastids or chloroplast are not present in animal cells. So centrosomes are related to cell division like mitosis and meiosis. So centrosomes forms spindle fibers. So centrosomes are only present at the animal cells and do not have presence at the plant cells. So B option is right. B will be the right answer. A plant cell lack a centrosome and lysosomes. Lysosomes are also not present at the plant cells. Which of the following polysaccharides is not present in the eukaryotic plant cell wall? Which polysaccharide is not present in eukaryotic cell wall, plant cell wall? Chitin, hemicellulose, pectin or cellulose? So cellulose, major or you can say most important thing in plant cell wall. Pectin, same. Hemicellulose, okay. But chitin is related to only animal cells. So chitin will not be present. Chitin will not be present in the plant cell wall. So yes, A option is right. Chitin is a polysaccharide that is present mainly on exoskeleton of arthropods and are not a component of plant cell wall. Which of the following molecules are the building blocks of a complex biomolecule such as protein, nucleic acid, polysaccharides and lipids? So proteins, for proteins we are having amino acids, okay. For proteins, amino acids, okay. So this is wrong, this is wrong, this is wrong, okay. Amino acids, right, okay. For nucleic acid we are having nucleotides, okay. So yeah, we are having nucleotides here. Polysaccharide is made up of monosaccharide units, okay. And lipids are fatty acid derivatives, okay. So A are right building blocks for the given compounds. So A will be the right answer. Protein is made up of amino acid. DNA is made up of deoxyribonucleotides. Ribonucleic acid, RNA is made up of ribonucleotides. Polysaccharide, that is glycogen, is made up of glucose, correct? Is made up of monosaccharide, that is glucose. Lipid, that is made up of fatty acid and glycerols. Next question we are having, which of the following cell organelle regulates the entry and exit of the molecules to and fro from the cell? So which organelle regulates entry? Lysosomes? No. If we are talking about the to and fro motion of compounds inside and outside the cell, so will it be Golgi bodies? No, not at all. Cell membrane, yes. It is the cell membrane that is responsible for the components, the compounds that has to be gone inside the cell or outside the cell. So it is cell wall that is responsible for the to and fro motion of compounds, entry and exit of the compounds in a cell. So cell membrane will be the right answer. Cell membrane regulates the entry and exit of the molecules from inside to outside of the cells and outside inside the cell. Which organelle contains DNA, the repository of genetic information? Which organelle have DNA? Is it Golgi apparatus, endoplasmic reticulum, mitochondria? No, not at all. It is nucleus. It's nucleus that contain the DNA. It's the nucleus. So nucleus is having double membrane organelle that contains the genetic material, and this genetic material is stored in the form of DNA. And the first person who discovered the nucleus was Robert Brown in 1831. Which organelle is the principal producer of ATP, the energy currency of the cell? Energy currency of the cell, ATP. So, is it Golgi apparatus, endoplasmic reticulum, nucleus or mitochondria? Okay, so this is again one of a very easy question. You can see that mitochondria are the principal producers of ATP via Krebs cycle. Mitochondria, the powerhouse of the cell, also known as. The mitochondria are the centers for metabolic oxidative reactions to generate and reduce coenzymes NAD and FADH2, which in turn are utilized in ATC to liberate energy in the form of ATP. Which organelle is responsible for the degradation of cellular compounds? Degradation of cellular compounds, is it Golgi apparatus that is required, that is basically used, that is responsible for the degradation of cellular compounds? No. It is lysosomes, yes. Peroxisomes, no, not at all. Endoplasmic reticulum, not at all. So it's lysosomes containing hydrolytic enzymes that are responsible for the degradation of cellular organelles. So yes, suicidal bags of the cell, lysosomes will be the right answer. Lysosomes are the digestive bodies of the cell actively involved in the degradation of cellular compounds. So tell me, what is the marker enzyme present in the lysosomes? This will be your homework. Just write down in the comment section. Okay, write it down in the comment section if you know. What is the main difference between a rough endoplasmic reticulum RER and smooth endoplasmic reticulum? Is it RER that contains ribosomes while SER do not? Yeah, bilkul. Is it true? Yes, RER contains ribosomes while SER do not have ribosomes. So that's why RER is responsible for the Protein synthesis. Protein synthesis. SER contains ribosomes? No, not at all. Both are here. Okay, wrong. A is the right option for this question. Rough endoplasmic reticulum RER is studded with ribosomes, giving it a granular appearance, while smooth endoplasmic reticulum SER do not have ribosomes. The largest organelle in cell is largest organelle. Is it endoplasmic reticulum, chromosomes, nucleus, or Golgi bodies? It is nucleus that is largest organelle. Nucleus that is having largest size. The largest organelle is the nucleus. The eukaryotic cells have a nucleus and genetic material is stored in the nucleus. The nucleus controls all cellular functions and is referred to as the control center or brain of a cell. The marker enzyme of lysosomes, the question that I have been asking you, the marker enzyme of lysosomes is, so is it acid phosphatase, alkaline phosphatase, lactate dehydrogenase, or a catalase? So it is acid phosphatase. Marker enzyme. What is it? Acid phosphatase. Lysosomes are spherical vesicles involved in a single membrane. Look at this last line. Acid phosphatase is considered as the marker enzyme of lysosomes. They are suicidal bags, and they are lysosomes enzymes may also destroy the entire cell that contain them, process known as autolysis. So autolysis is something that is related to the self-degradation of a cellular compound via lysosomes. Due to presence of hydrolytic enzymes, various digestive enzymes inside that lysosomes. Next question we are having, what is the jelly-like substance found floating inside the plasma membrane? What are those jelly-like substances that are floating inside the plasma membrane? Is it a cell sap, cytoplasm, karyoplasm, or mitochondria? So if you talk about floating substances found in a jelly-like substances, it is cytoplasm. So cytoplasm are enveloped by plasma membrane. This cytoplasm is basically made up of nearly 80% of the cytoplasm is having water content. It is made up of water, and it is the cytoplasm that is responsible for various organelles to organize that are present in that cytoplasm. Okay, so B option is right. B will be the correct answer, cytoplasm. It is present within the cell membrane. Cytoplasm of the cell is a jelly-like material formed by 80% of water. It contains a clear liquid proportional called cytosol and various particles of different shape and size. Okay, one of the most important question, that is the cytoplasm plus nucleus. Cytoplasm plus nucleus, they comprises what? What do you call it? That is known as protoplasm. So cytoplasm plus nucleus, they are also known as a protoplasm. Why is the biochemistry important in pharmacy? What is the importance of biochemistry? When we are studying pharmacy, what is the use of biochemistry? It studies the behavior of inanimate objects. Since it is the study of living cells, so how can you see that no, it is not inanimate objects study? So this is wrong. It helps in the synthesis of new molecules. So yes, biochemistry means study of chemistry with related to our living cells. So this study of biochemistry helps out in synthesizing new molecules like newer drugs and how these drugs are going to react with our living cells. So B will be the right answer. It studied the formation of rocks and minerals. No, not at all. Rocks and minerals. So it explores the behavior of celestial bodies. B will be the right answer. Importance of biochemistry in pharmacy. Drug synthesis helps designing and develop new molecules. Enzyme catalysis used for chiral drug production. Structure activity relationship, that is SAR, modifies drugs for better efficacy. Receptor ligand interaction ensures targeted drug action. Biopharmaceuticals enable synthesis of insulin, vaccines, and antibodies. Okay, next question we are having, ATS ribosomes occurs in. ATS ribosomes are present in the eukaryotic cell of animals. In eukaryotic cell of animal and plants, both eukaryotic cells and prokaryotic cells, all of the above. So ATS and 60S. We are having, ATS and 70S. ATS and 70S. We are having two ribosomal units. This ATS is made up of this ATS made up of 60S and 40S, while this 70S is made up of 50 and 30. 50S and 30S. This ATS is present in eukaryotic cells, either plant cell or animal. So both plant and animal cells are having ATS of ribosomal units, while this 70S is present in prokaryotic cells, and this 70S is made up of 50 and 30S. So eukaryotic cells of animal and plant both are having this ATS ribosomal unit. So prokaryotic cells are having 70S that are made up of larger subunit of 50S and smaller subunit 30S, while eukaryotic having ATS that is made up of 60S larger and 40S is smaller subunits. Okay, so they are the questions from this topic that are most important and that are also important for your MRB pharmacist vacancy. So try to practice them, try to figure out all the concepts behind them, and try to have maximum right answers from these questions. Try to practice all. So thank you so much everyone for watching our video, connecting with us. Thank you so much. We will be definitely meeting soon with some of the new concepts and new questions. Thank you all.